Welcome to Anatomy and Physiology II. My name is Dr. Rimkus. We begin with the functions of the nervous system. We see for the functions of the nervous system that there are three basic functions: sensation, integration, and response. When we look at sensation, we're talking about sensory perception, the way that we would bring in information from the surrounding environment. Once that information comes in, we need to decide what's most important. So we get an integration or a filtering of information. Finally, with all the information that comes in, we need to mediate the appropriate response. We need to send back a signal so that we can cause some muscle action. When we look at the organization of the nervous system, we see an interesting pairing of the system. We see the central nervous system, and then we also see the peripheral nervous system. And for each of these, we see that they are broken down into other pairs. So for the central nervous system, we see the brain and the spinal cord, and the peripheral nervous system, we see the afferent nervous system and the efferent nervous system. When we talk about the afferent nervous system, we're talking about sensory. So we're talking about information that would be coming in. When we refer to the efferent with the E, then we look at this in the terms of things that are moving out of the nervous system and the motor response. Further, we see that the efferent nervous system can be broken down into both a somatic nervous system as well as an autonomic nervous system. The somatic nervous system is the voluntary side, where the autonomic is involuntary. Let's explore these further. Looking at the somatic and the autonomic nervous systems, we can see some differences between those two. The somatic nervous system is comprised of the systems where we have conscious control of the movements. So conscious control of skeletal muscle. We decide to raise our hand. We decide to write notes. Beyond that, what we see is the somatic nervous system also has special neurotransmitters, or in this case, one particular special neurotransmitter, and that is acetylcholine. Acetylcholine is going to be used several times throughout this course, and in the future we may just refer to it as capital A, capital C, little h, ACH. In comparison, if we look at the autonomic nervous system, we see unconscious movements. Unconscious movements of things like smooth muscle, cardiac muscle, and glands. Things that we don't have direct control over. Now, we can have influence on these. Let's say I said, make your heart rate increase. Well, you could do that by thinking about some situation where you maybe got in a car accident or something, and that would cause you to start to envision yourself in that situation, and eventually you could see that you could get your heart rate increased because of that. So you could influence cardiac muscle, but it still is an unconscious effort in terms of the autonomic nervous system. If we look at neurotransmitters, the neurotransmitters within the autonomic nervous system are ACH, as we saw before, acetylcholine, but now we also include norepinephrine, another one that we'll see quite often throughout the semester, and that one we designate as norepi. Continuing on in the organization of the nervous system, we find now again another pairing or another level that we can go down where we find two other systems. So if we look at the autonomic nervous system, we can see that there is a parasympathetic nervous system and then also a sympathetic nervous system. Now, if we had to put some key words with parasympathetic, we would use the ideas of rest and digest. With sympathetic, we'd be looking at fight or flight. So running away or fighting and battling it out. Okay, let's pull all of this together. As you can see at the top here, we have the central nervous system. And underneath here, we have the peripheral nervous system. Within the central nervous system, we see sensory information coming in, we see an information processing, and then we see motor information that is leaving the central nervous system. If we go all the way down here, we see receptors, and those are the sensory organs within the skin and the outside of the body that are going to bring in information. So we have our information that's coming in. So we have special sensory information like for vision or smell, and then we have the tactile ones within the skin. All of those are bringing information in, so that is the afferent nervous system. So afferent is bringing information in, and again, efferent is bringing information out. So the sensory information is coming in, and then we have an opportunity to filter. So we have an information processing. So we have the opportunity now to decide what is the most important and what is extra material that could just be ignored. For instance, if I said, you put your socks on in the morning, and if all day you got signals that said, your sock is on your foot, 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 it would get irritating, annoying, distracting from the information that would be of a higher priority that would need to get through and you'd need to answer more quickly, such as your hand is on a hot stove. So if your hand is on a hot stove, that information would have to be higher priority than your sock is on your foot. Otherwise, your hand would definitely burn. And your sock would still be on your foot. So once we've done the information processing, then we can move on to the proper output that's coming out. So the motor command that would be leaving the central nervous system and going out into the peripheral nervous system. So again, if we look at the motor information that's coming out, we either have somatic or autonomic. So again, we see that two-branch system. If it is somatic, then it is a skeletal muscle and you're telling your hand to come away from the hot stove. If it is something that is happening within the autonomic nervous system, then we have to decide again between two branches, parasympathetic and sympathetic. So for parasympathetic and sympathetic, what we see is both of those are going to have as effector organs smooth muscles, cardiac muscles, and glands. Either way, the parasympathetic division or the sympathetic division will decide how those glands will react. In general, again, the parasympathetic is rest and digest. So if you're talking about something like cardiac muscle or smooth muscle, you would be thinking, oh, well, let's slow those down. But not necessarily. If the smooth muscle you're talking about is a digestive organ, you might be telling it to ramp up and increase digestion. So be careful with parasympathetic when it says rest and digest. Rest, yes, cardiac muscle, you're definitely going to slow down, but digest, your smooth muscles might be ramping up and increasing output or activity. Sympathetic is fight or flight. So for sympathetic, your cardiac muscles would definitely increase force and contraction. Now, your smooth muscles, if they were for digestive organs, might be completely shut off. All right, let's look at this all again, but now flip the whole diagram upside down. So now at the bottom we have the central nervous system going up to the peripheral nervous system, and then up at the top where we have our effector organs where the final response will be. So we start off with the stimulus. We see a bear. That's going to provoke a sympathetic response. Also, we can decide that we're going to run away, so there could be a somatic as well as an autonomic response. So we see the stimulus, we see the bear. That causes us to bring in information into the sensory neurons and into the peripheral nervous system. The peripheral nervous system is then going to convey that information to the central nervous system. So we have afferent coming in, then we have integration and processing happening within the central nervous system, where we're going to decide how serious of a threat this is. If this bear is 1,000 meters away, maybe not a threat at all, we can stop and take some pictures. But if we come upon this bear and we are within 100 feet or 50 feet, we might now think that we are in much more of a danger situation, and the integration would lead us to the motor response where we are going to decide to run away. Again, that's going to trigger two different responses, one on the autonomic side, and again, within the sympathetic, because it is going to be a fight or flight reaction, and then also on the somatic side, because we are going to make decisions, conscious decisions, to run away. So conscious decisions to run away, our effector organs are going to be skeletal muscles. Within the autonomic side, the sympathetic will tell us to use cardiac muscles, increase force and contractility, smooth muscles and glands, depending upon where they are in the body. Some of them within the digestive organs completely shutting off, other ones increasing output of adrenaline from the adrenal glands, etc. Okay, now we should look at the two different types of nervous tissue that make up the nervous system. We have neurons, which are the workhorses. These are the structures that are going to be carrying the action potentials. The neurons are going to have a very long lifetime. In fact, most of them are going to live every bit as long as you do. They are also amitotic. Amitotic meaning that if we look at the A in the beginning, that's from Latin words, A means without. The rest, mitotic, looks a lot like mitosis. So without mitosis. So that means that they are going to go their entire lifetime very early on, they'll divide, and then they'll stop dividing and never divide again. So they have this long lifetime, the ability to be amitotic, and they also have a high metabolic rate, and these are going to be the basic structural unit for the nervous system. The basic structural unit because they are the ones that are carrying the action potentials. Then we have neuroglia, and neuroglia, those are the supporting cells for the system. So let's take a closer look at a neuron, and then we'll move on to neuroglia next. So for the neuron, what we see is a cell body, and the cell body will have processes coming off of it. The cell body is sometimes referred to as a soma, S-O-M-A as well. So for the cell body, we have a nucleus, and then we have these processes that are on the outside. So we see these processes, and over here we see these called dendrites. So the shorter ones on the outside are generally referred to as dendrites. And then there is one or more very elongated process that is referred to as an axon. So here we see one major axon leaving, and then at the end of the axon we have these axon terminals at the end. Along the length of the axon, we also see these myelin sheaths. So the myelin sheath is a Schwann cell, and in particular, that Schwann cell is referred to as a neurolemmacyte. But we'll see those as we get into the different types of neuroglial cells. Let's go back to the dendrites and how they're working. The dendrites will form a receptor field around the cell body. They are going to be drawing information in. So information is coming in via these dendrites, being processed within the cell body, and then moving down the axon to the axon terminals, where the axon terminals will have chemicals that will leak across. Now going back to these neurolemmacytes, we see that there are little gaps, and those little gaps are referred to as the nodes of Ranvier. So we have neurolemmacytes This is a myelinated nerve fiber. If they were absent, you would have an unmyelinated nerve fiber. As we can see, there are three different classifications for neurons. They can be sensory or afferent neurons, ones that would be bringing information into the nervous system, into the central nervous system. And then we have interneurons, which would be inside of the central nervous system. They can also be called association neurons. And these, again, are the ones involved in the filtering component or the decision-making aspect. And then we have the motor neurons, the ones that are going to go back out to the peripheral nervous system. So there is a nice parallel here between the three functions and the three different types of neurons: sensory, integration, response. Sensory neurons, afferent neurons, interneurons or association neurons on the integration side, and motor or efferent neurons on the response or the motor side. Next, we'll run through neuroglial cell types or neuroglia types. For the neuroglia, we see astrocytes, ependymal cells, microglia, oligodendrocytes, neurolemmacytes, and satellite cells. For each of these, they are supporting the neurons. So neuroglia will not be carrying action potentials, but will be providing an environment where the neurons can function properly. Let's take a closer look at these 6 different neuroglial cell types. First, we have the astrocytes. The astrocytes get their name because they are shaped like a star. They have cytoplasmic extensions that come off in all different directions so that they can wrap blood vessels or neurons themselves, and even lining the pia mater. They are going to help regulate the fluid composition or the extracellular fluid that is around the neurons. So you can see, as in this case, they are wrapping capillaries, which would be leaky, and they're preventing certain fluids from coming out so they can provide the proper environment for the neuron to work. Next, we see the ependymal cells. The ependymal cells, as we can see, come in 2 different types. These have cilia along the top, and these are more of a glandular type appearance. The ependymal cells are responsible for producing cerebrospinal fluid, or CSF. Now, we see the glandular ones, they're responsible for the actual production of the cerebrospinal fluid. And then we have the ciliated ones, which would be responsible for the movement or the circulation of the cerebrospinal fluid. These ependymal cells are going to line the ventricles of the brain and the central canal of the spinal cord. Therefore, those are the locations where they can release the cerebrospinal fluid and then also use the cilia to circulate the cerebrospinal fluid. Another neuroglial cell type is microglia. Microglia are tiny little cells. Tiny little cells that are able to transform into a mobile phagocyte. Mobile being able to move, a phagocyte something that is going to surround and destroy materials or other cells, and they are transforming in response to inflammation, the presence of microbes, or other foreign substances that are trying to invade the central nervous system. The microglia are also responsible for cleaning up dead tissue or necrotic tissue deposits. We also see oligodendrocytes as a type of neuroglial cell. And here, within the oligodendrocyte, what we see is the formation of cytoplasmic extensions, but this time the cytoplasmic extensions wrap the axons. Oligo means few. So the oligodendrocytes are going to wrap a few axons that are running adjacent to each other within the same area. When we have one myelin sheath that begins, and then a gap, and then another one that is on either side, the gap again is called the node of Ranvier. So we see myelin sheaths produced here as well, and again we're wrapping 3 or 4 adjacent axons. All of these axons are within the central nervous system, where we very typically have axons that are all running in the same direction in the same area. So the oligodendrocytes are going to wrap a few axons that are nearby each other within the central nervous system. Another type, the neurolemmacyte, is found within the peripheral nervous system, and here we see a neurolemmacyte will only wrap one axon, and again it's within the peripheral nervous system instead of the central nervous system. So to think about this in terms of an exam or what you should know, we have neurolemmacytes. They are present within the peripheral nervous system. They are wrapping one axon, and they are a neuroglial cell type. If we went back to the oligodendrocytes, we would see that they too wrap axons with myelin sheaths. But the difference is they're within the central nervous system, and they are wrapping a few axons that are running in the same direction. So if I was going to ask you a test question, I would want you to be able to differentiate between those two types of cells. Finally, we have the satellite cells. The satellite cells are found within the peripheral nervous system, and they are surrounding cell bodies within the peripheral nervous system, keeping the information that is within this cell body in this cell body and not letting it get short-circuited over to another cell body. So these satellite cells are insulating cell bodies. When we're within the peripheral nervous system, we have areas where we cluster the cell bodies. Clusters of cell bodies within the peripheral nervous system are referred to as ganglia. Again, ganglia are clusters of cell bodies. If we're only talking about one cluster, it is a ganglion, O-N at the end instead of the A.